Welcome back. So in the last few videos we've been talking a lot about mitosis, the regulation of mitosis, using things like cyclin-dependent protein kinases and cyclin and all that stuff. And we came to a conclusion. And the conclusion we came to at the end of the last video was that cells don't like mutation. They would like to theoretically do anything possible to prevent DNA from being mutant. And so as a result of this, we came to two truths. The first is that if the mutation in the DNA is minor, essentially what will happen is the cell, during the cell cycle, will prevent entry into the S phase, and during that halt in the activity of the cell cycle, the cell will attempt to repair its DNA using the DNA repair enzyme machinery it has in the nucleus. The other truth we elucidated was that if the cell in any way deems that the damage to the DNA is too great, then what's essentially going to happen is the cell is going to commit suicide. Okay? Now, there's basically two ways that the cell can die. It can either die through something like necrosis, which is a non-programmed type of cell death, but in this case, it's going to be a programmed cell death, and it's referred to as apoptosis. Okay? And we're going to talk about the molecular mechanisms of apoptosis in this video. And just to give you kind of an intuition of why you would want to do this. See, these cells are going to divide unless they're mutated, in which the cell will commit suicide. If you think about what would happen if every cell automatically, if you think about what would happen if every cell had to automatically divide and there was no regulation of it, is that if you have mutation, maybe improper base pairing in the DNA, some kind of covalent modification to the DNA that you don't want. If that were the case and you had no regulation of it, then for the most part, every one of those mutations in the DNA would be given to the future generations of that cell because you're not repairing it. So the idea behind apoptosis is that if in any case the DNA damage is too great, the cell will just kill itself and prevent future generations of cells being made that have the mutant DNA. And in this video, we're going to talk a lot about the mechanisms of apoptosis. And in general, when we talk about apoptosis, we're talking about a particular type of enzyme known as a caspase. Apoptosis, quite honestly, is one of probably the most underappreciated and neglected topics at the undergraduate level. Caspases, essentially what they are, is they're enzymes that promote apoptosis. If you really wanted to get technical as to what a caspase is, it's called a cysteine. It's called a cysteine protease. Likely, if you've taken any kind of biochemistry course, you've talked about serine proteases. Mechanistically, these are very similar, except in serine proteases, the nucleophile is serine. Here the nucleophile is cysteine, and other than that, the mechanism is basically identical from an organic point of view. If you're looking at the origin of the name caspase, what it essentially is it's a C-terminal aspartate protease. And you basically take several letters, number one, the C for C-terminal, asp in aspartate, and then ase for protease. And so what you get is caspase. So literally what caspases are, from a molecular enzymatic point of view, is they're basically proteases. Well, they are proteases. And so if you have a setup like this, like for example, let me do this. Let's say that I have a hypothetical sequence of amino acids. So let's say this particular residue right here, let's say that's an aspartate, right? And then here is the C-terminus of that particular residue. And then it just goes on like that. So literally what caspases do, it's a little bit more complicated than this, but what they essentially do is they look for aspartate residues in particular proteins. So it turns out that this residue that I've shown right here, this is an aspartate residue. It has one carbon here and then a carboxyl. So the caspase will look for that, and it will essentially, hydrolyze the peptide bond on the C-terminal side of that alpha carbon. So the alpha carbon I'm concerned about is this one right there. So it's going to cleave the C-terminal peptide bond, and that happens to be this one. Okay, so that's the bond that gets hydrolyzed by the caspase. It's the C-terminal amide bond. Okay, so mechanistically, that's what they do. In another video, we'll actually look at the full-fledged organic mechanism. Okay, enough about that. Caspases are synthesized as zymogens. So what is a zymogen? A zymogen is an inactive enzyme. And we'll find there's actually good reasons why you synthesize them as a zymogen. If you ever want to activate the caspase, you're going to have to cleave off part of the protein. We'll talk about that in just a minute. If you had fully active enzymes, fully active caspases that were just made like that, all you would do is induce apoptosis, right? Because if you make the active enzyme, it's immediately just going to catalyze its reaction and the cell would die. So you're only going to pull out the caspase active machinery when you need it. Other than that, it's synthesized in an inactive zymogen form, and it has to be activated. And we're going to look at the mechanism to see how that occurs. Basically, with caspases, the source of their inhibition is what we call an inhibitory peptide. So basically, for all intents and purposes, there are two parts to the caspase. Okay, there's what we call a fully active catalytic component. That's what does everything. And then there's an inhibitory peptide. Those are the two main things we're concerned about. In the inactive form, you have this inhibitory peptide that is covalently attached to the caspase. Okay, and if you ever want to activate the caspase, there has to be some mechanism to cleave off that inhibitory peptide, and hopefully that makes sense. So it has to be hydrolyzed off. Okay, in general, this is the truth right here. The removal of the inhibitory peptide converts the procaspase into an active caspase. Okay, so whenever you're naming caspases, if it's in the zymogen form, meaning before it's been activated, you call it a procaspase. And then whenever it's in the fully active form, you simply refer to it as caspase. And there are multiple caspases. There's about 10 main ones that are of really critical importance, some of them more important than others, and you designate which one you're talking about by putting a number after procaspase or caspase. So for example, these caspases that are shown over here, okay, these ones over here, these are the ribbon diagrams for caspase 8. Caspase 8, as we'll find, is what we essentially call an initiator caspase. There are also executioner caspases, okay? So what we're going to talk about right now is something referred to as the extrinsic pathway of apoptosis. The reason it's called extrinsic is because basically in this case the signal for destruction of the cell comes from outside of the cell. Okay, and in general, there's two things that have to be present for this to occur. Number one, in general, there has to be something referred to as a Fas receptor. Okay, sometimes they'll simply refer to this as just Fas, F-A-S, but understand that this molecule we're talking about is an actual receptor, what's shown basically in tan right here, and it goes ultimately through the membrane into the cytosolic side. Okay, also there are these ligands called Fas ligands, and these are basically the ligands of the Fas receptor, and so you designate those FASL, or we can write it out Fas ligand or ligand, however you want to pronounce it. Okay, what ends up happening is the Fas receptor on the cytosolic side, okay, it has these extensions, these you could sort of consider them almost like a coenzyme attached to it. There are these subunits that kind of come off of the cytosolic side. These are called Fas-associated death domain. Let me highlight that. Fas-associated death domain. And what the death domains of the protein complex essentially do is they're going to activate one of the initiator caspases. Okay? So what you essentially have is you have this Fas receptor in the membrane in here, and then on the cytosolic side, you have these Fas-associated death domains. The Fas-associated death domains are going to bind an inactive caspase known as procaspase 8. Okay, so what's going to happen is there's going to be a Fas ligand that generally comes from a cell like a cytotoxic lymphocyte. There are different immune cells like cytotoxic lymphocytes, natural killer cells. These are both immune cells that induce apoptosis, and generally the way they do it is through the Fas ligand. So imagine there's a cell up here like a lymphocyte, some kind of immune cell, maybe a lymphocytes up here that's donating the fast ligand. So it's a little bit kind of a misleading picture. But imagine that some immune cell of some sort is donating the fast ligand, and when the fast ligand binds to the fast receptor, it causes trimerization of these receptors. And that brings, of course, 3 fast-associated death domains together. And when they're together, they're able to basically take pro-caspase 8 and activate it into the fully active, let me do this in light blue, caspase 8. Okay, so like I said, whenever we activate a caspase, we're taking it from a pro-caspase into a caspase by cleaving off the inhibitory peptide. So pro-caspase 8 loses the inhibitory peptide to become caspase 8. Caspase 8 is what we call an initiator. This is called an initiator caspase. And in general, the reason we call it an initiator caspase is because essentially what happens is it's the first one that's activated in the pathway. We're going to also run into another one called caspase 9, which is also an initiator caspase. The initiator caspases in general don't do any of the dirty work. Okay? The dirty work is done by these, they call them effectors here, but these are actually called executioner. That's the common name. Executioner caspases. So the executioner caspases are essentially what get their hands dirty. They're the ones who end up activating DNA hydrolases. They proteolyze cytoskeleton elements. So the guys that are actually doing the dirty work, getting their hands dirty, those are the executioner caspases. And in general, we have 3 of them. We have caspases 3, 6, and 7. Those are the main ones. Okay, there are some others, but those are the 3 main ones that anybody should be aware of. Caspase 8 is an initiator caspase because what caspase 8 does is it activates these 3 guys. So caspase 8 activates caspase 3, it activates caspase 6, and caspase 8 activates caspase 7. Okay? So literally, the initiation of the apoptotic pathway is done by caspase 8, but the actual execution of the cell, or on some level you could consider execution of the orders, that's done by caspases 3, 6, and 7. Okay? And that's basically all there is to the extrinsic pathway. You have some cell that kind of floats by the cell that it wants to kill. The cell that needs to die is expressing fast receptor, and so a fast ligand is donated to the fast receptor. You get activation of caspase 8, and that executes activates the executioner caspases, and that leads to the apoptotic response. Okay, why don't we go ahead and kind of fill in what the apoptotic response is? So executioner caspases are activated by initiator caspases. Okay. Also, in some cases, executioner caspases can actually be activated by other executioner caspases. So it's like the snowball effect when you roll it down the hill. Once you really start activating the initiator caspases and some more executioner caspases, you really start activating these guys more and more and more, and it's ultimately going to lead to cell death. Okay. In general, the executioner caspases we're concerned about are numbers 3, 6, and 7. One of the things they do is they activate an enzyme called a DNase. Okay. What a DNase is essentially is it's an enzyme that breaks apart DNA strands. Okay. Basically, what you have in the DNA, this is not something that's required for the most part, but you'll have these proteins. Okay. There are these proteins. And these proteins are called histones. Okay? And essentially the DNA, the way it works, is the DNA sort of wraps itself around these histones, and that's part of the DNA structure. The DNase, sometimes they'll abbreviate it like this, DNase, it likes to clip right here, it likes to clip right here. Basically, it clips between the histones. Okay, that's really characteristic of the DNase that gets activated by these caspases. It will literally clip between the histones, but it will ultimately clip the DNA into individual pieces, okay, like this. So the DNA gets cleaved apart. The remnants of the DNA are packaged into basically vessels called apoptotic bodies. Okay, apoptotic bodies, they sort of branch off from the dying cell, and they end up getting phagocytized by immune cells like macrophages and things like that. Okay, so basically the way you can sort of view it is you have a dying cell, right? a dying cell, okay, and it has some material that's eventually going to get budded off, so you'll form something like this. You'll have some material in there, and then it'll eventually come off. And this thing right here, this is the apoptotic body. Okay, this is the apoptotic body, and eventually what's going to happen is some kind of phagocyte, like a macrophage, is going to engulf it, phagocytize it, and all the contents of it are basically going to be recycled. They're just basic molecules that float around the cell, so we'll basically just recycle them. So that's kind of one of the beautiful things about apoptosis, is it's not a total waste. Some cells just have to die. Okay, they're old, they might be mutated, they have to die, but we can recycle the things that are inside them. Another thing that Caspases 3, 6, and 7 do is they degrade cytoskeleton elements. Okay, so part of the cytoskeleton of cells are the microtubules, the intermediate filaments, the microfilaments, all sorts of different branches of those things. You can get really specific. In general, they degrade those cytoskeletal elements along with keratin and actin. Okay, so the idea is that anything that gives the cell structure, these Caspases are going to target for destruction. Okay, there are also some nuclear proteins that are also degraded by the executioner Caspases. Okay, so the main idea behind the Caspases is destroy anything that gives the cell function, so the cell will die, and eventually all of the components of the cell will essentially be removed through apoptotic bodies. They'll get phagocytized and recycled. Okay, so hopefully that makes sense. And I mentioned in this slide we were talking about the extrinsic pathway. The reason this is called the final common pathway is because there's also an intrinsic pathway that converges with this, and that's actually what we're going to talk about right now. Before we get into the intrinsic pathway, I have to make sure you understand something. Okay. The intrinsic pathway is intrinsic because it's stimulated by things inside the cell. So different cells have different lifespans. Some cells don't live very long, in which case the apoptosis occurs rather quickly. Other cells actually live a lot longer, and so apoptosis will be triggered a lot later. And the control of whether or not a cell goes into apoptosis, in large part, is accomplished through genes. Okay? So here's what we have. We essentially have these things called pro-life genes. A pro-life gene is something that promotes cell survival. In other words, it's anti-apoptosis or anti-apoptotic. Two of these proteins that you should be aware of are number one, BCL2 and BCLX. Usually those members of the BCL family, okay, those are your pro-life genes. And so when they get transcribed and ultimately translated, their protein products, BCL2 and BCLX, are also pro-life. Okay, and then you have these other guys which are pro-death. Okay, so these are your pro-death or pro-apoptotic genes, and so their proteins are bad. So number one, we have A, which is bad, B, which is backs, and C, which is back. If you remember, they all sort of sound like bad. If it's pro-death, it must be bad, right? Not a very good analogy, but hopefully that gives you a little bit of intuition. Bad, backs, and back. Those are your pro-death proteins, okay? And really, sort of the ratio of the pro-death proteins to the pro-life proteins, in large part, sort of determines the path that the cell's going to go. Is it going to be promoting cell survival, or is it going to promote apoptosis? Okay, so here's the idea. In general, if you have a lot more pro-life proteins than pro-death proteins, that's going to promote cell survival. If you start tipping the balance a little bit and you start making more pro-death proteins, that's what triggers apoptosis. Okay, and like I said here, the relative amounts of each group of proteins determines the fate of the cell. So here is probably the most critical thing to understand about this slide. Okay, these proteins that get made, they're good at dimerizing. Okay, so you can get a pro-life, pro-death dimer. Okay, you can get a pro-life dimer, and then the last kind you can get is you can get a pro-death dimer. Okay, and I've sort of given you the possible arrangements you can have. I think if you look at it, it'll make a little bit of sense. If you have a pro-life and pro-death dimer, that means you have a pro-life protein dimerizing with a pro-death protein. So an example of that would be, you take this one, BCl2, and dimerize that with BAD or something like that. Okay, in fact, I do use that one. Here it is. Pro-life, pro-death dimer is BCl2 and BAD. Okay, and you can mix and match these as much as you want. Okay, pro-life dimer. Okay, that would be like BCl2 and BClX. I have that example right here. So what do we have? We have BCl2, let's see, BCl2, BClX. Okay, so you can have all sorts of pro-life combinations, basically. Okay, I didn't include that one, but notice here's BCl2, BCl2. Okay, so the idea is that they dimerize. You could also have a pro-death dimer. Okay, that would be like BAD and BAX. Okay, here's the example. Let's see, BAD. Here's BAD and BAX. Okay, so hopefully that makes a little bit of sense. They dimerize. Okay, now here's where that comes in handy to know. The mitochondria, we have this big idea that it produces energy. Right? That's where the respiratory chain is. That's where the TCA cycle is. Energy must come from the mitochondria. But you can look at things that are really, really, really good, but there's always a catch. And the catch with the mitochondria is even though it produces so much energy, it has these pores. Okay? The mitochondria effectively has these holes. Okay? And so what essentially will happen is these dimers, when they come together and dimerize, they like to get in front of these pores. Okay? So the idea is that when you have a pro-life, pro-death dimer, that's good at blocking the hole, these pores. It's good at blocking that. Okay? If you have a pro-life dimer, once again, it's good at blocking that pore. Here's the problem. When you have a pro-death dimer, it turns out that the structure of that dimer is really bad at blocking those pores. So this guy right here, the pro-death dimer, really bad at blocking those mitochondrial pores. So why would that be bad? Well, inside the mitochondria, what do we have? Well, in the respiratory chain, pretty much every enzyme and protein in there is in the membrane, right? It's in the inner membrane of the mitochondria. But if you remember back to your physiology, there's one particular protein in the mitochondria that is soluble, and that's cytochrome C. Very important to realize that cytochrome C, it turns out, because it's soluble, it can actually try to leak through these pores. So if you start building up a lot of pro-death dimers because you're making more pro-death proteins, then the actual amount of these guys right here, the pro-life, pro-death dimer and the pro-life dimer, these guys go down relative to the pro-death dimer, which actually go up. So you basically start making it harder and harder to block those pores, and you make it more likely for cytochrome C to escape the mitochondria and end up in the cytosol. And that's where this apoptosis is going to be triggered. So what you should realize is that as you start making more pro-death proteins, you get more pro-death dimers, and therefore mitochondrial pores can't be blocked as well, and cytochrome C comes out. Now, cytochrome C likes to bind with this protein called APAF1. What APAF1 essentially is it's called apoptotic activating factor 1. Apoptotic activating factor 1, but most people just refer to it as APAF1. So cytochrome C is this red circle right here. You can kind of see cytochrome C here existing in a complex with APAF1. And in a nucleotide-dependent fashion, so you can actually either use deoxy-ATP or ATP, but basically in a nucleotide-dependent reaction, which is ultimately going to be a nucleotide hydrolysis, what you allow for is the polymerization of these subunits. What am I talking about? This subunit right here. This is a monomer. Okay, this right here, this is a monomer. This is the monomer of something referred to as the apoptosome. Okay, so you basically take 7 of these monomers, and by the way, the monomer is APAF1 bound to cytochrome C. Okay, you take 7 of those and you polymerize them into basically a protein complex known as the apoptosome. And what the apoptosome does is it activates a caspase, and the particular caspase it activates is called procaspase 9. So basically, the apoptosome activates procaspase 9 to simply caspase 9. And this is, of course, the activated form of the caspase. So you know what the apoptosome does is it triggers the hydrolysis off of the inhibitory peptide, and now you have activated caspase 9. Now you have a very highly activated apoptosome, and what it's able to do is activate procaspase 3 into active caspase 3. And not only is able to do that, but it will also react with caspases 6 and 7, so those get activated. So also bear in mind that if you activate caspase 3, you also get activated caspase 6, and you also get activated caspase 7. And those three right here, these guys right here, these are your executioner. These are your executioner caspases. They are what are doing the deed, right? So if I go back to this slide, the final common pathway that starts with executioner caspases 3, 6, and 7, they're the ones who are activating the DNases that degrade DNA by clipping between the histones. They're the ones that degrade the cytoskeleton elements like microtubules, intermediate filaments, and microfilaments. And they're ultimately the ones that trigger the destruction of nuclear proteins, and you end up getting apoptotic bodies that basically destroy the cell, and you get recycling of the parts. So hopefully that makes a little bit of sense, and I already went over this basically in the last slide. But understand that cytochrome C binds with apoptotic activating factor 1 or APAF1. Those monomers get polymerized into an apoptosome. The apoptosome binds and activates procaspase 9 to caspase 9, and then that highly activated caspase 9 activates procaspase 3 to caspase 3, and then you also get activation of caspases 6 and 7. And those final three, 3, 6, and 7 are the executioner caspases. Okay? One little application of this that you would probably see in an immunology class or anatomy and physiology too is you talk about these cells called cytotoxic lymphocytes. These are very, very dangerous cells. Okay? They're part of the specific immune system response. And here's the idea behind a cytotoxic lymphocyte. The way that I was taught about them is they're basically the sadists of your body. They're literally sadistic. So if you think about like a horror movie or so of some kind, basically what a sicko would do in one of those horror movies is they would literally take a drill. They take a drill and they drill it into your side. So there's literally a hole in your side. And then if that wasn't enough, then they take some kind of noxious chemical and then they dump it into that wound, right? Terrible thing. But in fact, on a molecular basis, that's exactly what cytotoxic lymphocytes do. So what they do essentially is they're the ones, number one, that have fast ligand. So they're able to use fast ligand to induce apoptosis through the extrinsic pathway. But it turns out they're a little bit more sadistic than that. So what cytotoxic lymphocytes do is they make a protein called perforin, or perforin. This perforin protein, or peptide, likes to polymerize. And so you can actually see the result of its polymerization right here. So essentially what happens is when perforin polymerizes, it likes to polymerize in the membrane of the target cell, which is shown right here. Okay? When the perforin polymerizes, it kind of makes a hole, and the hole allows things to go into the target cell. For example, what the cytotoxic lymphocytes like to do is they like to dump in enzymes known as granzymes. Granzymes are serine proteases. So a granzyme is a serine proteases, and what it likes to do is activate executioner caspases 3, 6, and 7. So not only did the cytotoxic lymphocyte drill a hole in the target cell, it also dumped in some granzymes which activate the executioner caspases that induce apoptosis. And if that wasn't enough, of course, it also uses the fast ligand and activates the extrinsic pathway for apoptosis. So cytotoxic lymphocytes, very dangerous. They'll kill anything they come in contact with as long as they're specific for it. Another type of cell that's part of the innate immune response, that's mostly responsible for immunological surveillance, responsible for killing of cancer cells. I misspelled it here, but you can kind of get the idea. Natural killer cells, they're part of your innate immune response. They also do the same type of thing. Cytotoxic lymphocytes, they're a little bit more sadistic. Whenever they do it, they will literally poke holes using perforin, dump granzymes in. Natural killer cells, they don't do that, but they do use the Fas ligand. So natural killer cells do induce apoptosis, but they only do it through the Fas ligand. So natural killer cells, they use the Fas ligand. Okay, but cytotoxic lymphocytes can do it both ways. So hopefully this video gave you a little bit of intuition on apoptosis. And just keep in mind what we talked about at the very beginning. The reason you have to have this is because if you have DNA in a cell that's damaged, the last thing you want in the world is to replicate the DNA and pass that genetic information that's mutant along to future generations of cells. You have to realize these cells, you have a finite number of cells in your body, and if you want to make any more of that type of cell, it has to ultimately come from a previous cell that divided. So you don't want cells that have mutant DNA. That's not a good thing from a biological perspective. So you prevent further mutant generations of cells by killing the cell before it gets a chance to replicate. So hopefully that made a little bit of sense. See you in the next video.